All right, this lecture is continuing on with chapter DNA. We just talked about DNA structure and replication, that beautiful process. It's going to duplicate our DNA during S phase, interphase. Now we're going to continue on with the central dogma when we are going from genetic material to protein. And so the different processes that are involved in that are transcription and translation. Let's go ahead and get to it. Okay, first poll. Which DNA strand is complementary to the strand of DNA? Looks like I'm going to need 3 prime, GATT, CAC, AT, 5 prime. So this is callback from the previous lecture and previous units as well, knowing how bases are going to pair up as its complement. G over here, I'll talk about the primes in a bit, but G will be bonded, hydrogen bonded with C in DNA, A with T, T with A, G, T, G, T, A. And so it looks like I'm giving you those options here. Here's that strand, just looking at the letters, and it's the same over here. So it's kind of the same question where I'm not really too concerned with you guys knowing about base pairing because you show me that you can do that beautifully. It's really those prime ends and knowing that complements are anti-parallel to each other. So here's the 3 prime over here. If it starts on the 3 prime on the left side, then its complement must start on the 5 prime of the left side. And answer A is showing that. Same on the other side, whereas the original strand has 5 prime. Remember that a phosphate functional group is going to be found on that side. Then its complement, anti-parallel, kind of opposite if you will be 3 prime. Then a hydroxyl group will be found on that end, hanging off the sugar. A is a correct response. So we've looked at a DNA nucleotide in great detail. Just see that I'm recording. I am. Okay. So now let's go ahead and bring back the RNA nucleotide. Remember, a nucleotide is a monomer for nucleic acids. I'm going to look at the differences between a DNA and an RNA. You guys have seen this slide before. Let's recap. Number 1, there's still a sugar in RNA nucleotide. That's true. It's a different sugar. It's ribose instead of deoxyribose. Difference number 2, instead of thymine in RNA, that's replaced by the nitrogenous base uracil, uppercase U. So we have A, G, C, and instead of saying T, we say U now for RNA. Difference number 3, it is usually found in the single-stranded form. DNA in eukaryotes is double-stranded. It has a strand, and then that strand will have a complement, so it is a double-stranded helix. In RNA, you can be either double-stranded or single-stranded. In fact, it's mostly found in single-stranded form in eukaryotes. So this is your central dogma. As you guys go through this lecture, I really want you to go back to this image, write it down, draw it in your notes, because this is really what we're talking about, the different steps to go from what to what. And we are going to be discussing transcription and translation today. So let me get my pointer. This is the flow of genetic information going from DNA to protein. So going back, we talked about inheritance. So we have DNA. There's my double helix, double-stranded. A certain segment of the DNA, a certain part, not all of it, a certain part, in fact, very few parts of your DNA, is going to code for a gene. Genotype, we discussed, is the heritable info contained in that sequence of nucleotide bases in DNA. Those nucleotides, those monomers, and a different number of them, are going to code for a gene. That's what gets passed down from parent to offspring, and that is what's going to give you the different traits that you have. Let it be red hair, freckles, like me. All right. So DNA provides instructions. It's a blueprint for a cell. What do those instructions do? We'll continue on with our central dogma to discover that. Transcription is the first process of this lecture that I'm going to discuss. Appreciate that transcription is a nucleic acid language. So we're going from nucleic acid to another nucleic acid, RNA. We're going from DNA to RNA. We're still talking about the nucleic acid language. That doesn't change. We're still in that biological molecule, nucleic acids. It's just changing from DNA to RNA. You're transcribing something into the similar form of language, nucleic acid, but it is a little different. Appreciate that this process occurs in the nucleus, going from DNA to RNA. RNA then is going to convey information over to a ribosome that acts as a messenger, in addition to doing some other things too, but we're really talking about mRNA. That'll come up in the lecture. So this is acting as a messenger. And then the process of translation will occur in the cytoplasm, outside of the nucleus. Translation is now going from nucleic acid language to protein language. Completely different language. Think of a translator going from English to Spanish. You're translating different languages. And so that's what's occurring here very much in our cells. Going from nucleic acid, specifically RNA, messenger RNA, to protein language. That is called translation. It occurs in the cytoplasm. Proteins then, why make a protein? Why go through all those steps? Well, that's going to give us phenotype. We're going from gene to an actual action of proteins. How you're going to look, what jobs your cells are going to do, the workers of our cells, proteins.